1 in every 4 people alive today lives in a country that was once part of the British Empire. That's not a historical footnote. It's a genetic fact, still measurable in living bloodlines on every inhabited continent, in populations that have never set foot on a British island. A woman in Sydney, tracing her family through 5 generations of Australian ancestors, finds her DNA converging on a small parish in Yorkshire she's never heard of. A man in Barbados with a surname that sounds unmistakably African discovers a Scottish lowland signature woven through his genome that predates emancipation by more than a century. Neither expected it. Neither had a family story to explain it. So geneticists asked a question no textbook empire map had ever really answered. If the British Empire eventually governed roughly a quarter of the world's land and population, how far did the genes of the people who ran it actually travel? Not the flag, not the language, not the legal system, the DNA itself. The answer required something no historian working from paper records alone could have assembled. It required comparing modern genomes from 6 continents against a detailed genetic map of the British Isles themselves, tracing exactly where colonial-era British ancestry ended up and how much of it survives quietly in populations that have spent centuries building entirely separate national identities. Start with the source material, because it matters more than most people realize. The British Isles aren't genetically uniform, and they never really were. A landmark 2015 study published in Nature, led by geneticist Peter Donnelly and colleagues from the Wellcome Trust Center for Human Genetics, analyzed DNA from more than 2,000 people across the United Kingdom whose grandparents had all been born within a few miles of each other. The goal was to reconstruct as precisely as possible the genetic geography of Britain before mass internal migration, urbanization, and 2 world wars blurred it beyond recovery. What that study found surprised even the researchers. Britain wasn't one population. It was a patchwork of at least 17 distinct genetic clusters corresponding closely to old regional and even tribal boundaries, some of them tracing back more than 1,000 years, to Anglo-Saxon, Norse, and Celtic settlement patterns that modern county lines had almost, but not quite, erased. Orkney showed a distinct Norwegian genetic signature from Viking-era settlement. Wales and Cornwall retained older Celtic-associated ancestry, less diluted by later Anglo-Saxon migration. Southern and eastern England carried the heaviest Germanic influence, a legacy of Anglo-Saxon settlement in the centuries after Rome withdrew. Perhaps the most unexpected finding of that study was how little the population had actually moved internally for most of English history. The genetic clusters mapped so closely onto medieval kingdom boundaries, some dating back to the Anglo-Saxon heptarchy of the 7th and 8th centuries, that researchers could sometimes guess a person's ancestral county to within a few dozen miles based on genetics alone, a level of local stability that surprised historians and geneticists alike. It meant that until the industrial era finally sent large numbers of Britons moving toward cities and eventually overseas, most communities had remained rooted generation after generation on the same patch of ground for well over 1,000 years. That internal diversity matters because it's the baseline researchers needed before they could trace anything abroad. Without a fine-grained map of the source population, comparing colonial descendants against British ancestry as a single category would have been almost meaningless, like trying to identify a single voice in a choir without knowing the individual singers first. It also meant that when a distinctive regional signature turned up in Sydney or Bridgetown or Toronto, researchers had a real chance of tracing it back, not just to Britain, but to a specific county, sometimes a specific parish, the way a fingerprint points to one individual rather than a general description of a hand. With that map in hand, researchers turned outward to the places British colonization actually sent people, and the genetic record turned out to be far more extensive and far more selective than the history books had prepared anyone to expect. Australia is where the pattern shows up most starkly because the colonial population there began under conditions almost no other British settlement shared. Beginning in 1788, Britain transported more than 160,000 convicts to Australian penal colonies over 8 decades, alongside free settlers who followed once the economy stabilized. Modern genetic studies of Australians with deep colonial ancestry show a population overwhelmingly dominated by British and Irish genetic signatures, with English ancestry from southern and eastern England particularly concentrated, alongside a substantial Irish component reflecting the disproportionate number of Irish convicts transported under British rule following repeated famines and uprisings. What's striking is how specific the regional signal remains. Researchers comparing Australian genomes against the fine-grained British reference map have found that descendants of convicts transported from specific English counties still carry detectable genetic signatures tied to those exact regions more than 2 centuries and 8 or 9 generations later. A crime committed in a Yorkshire village in 1820 could still be legible in outline in an Australian genome in the 21st century, not through any record or family story, but through DNA that simply never stopped being passed down. Australia's convict population itself skewed toward specific crimes and specific regions, which further sharpens the genetic signal. The overwhelming majority of transported convicts had committed property crimes, often minor theft driven by poverty in Britain's rapidly industrializing cities rather than violent offenses. London, Yorkshire, and Lancashire, 3 regions hit especially hard by the economic upheaval of early industrialization, supplied a disproportionate share of the convicts who eventually became Australia's founding colonial population, meaning modern Australian genomes with convict ancestry often trace back not to Britain broadly, but to the specific urban and industrial counties where economic desperation was most acute in the late 1700s and early 1800s. Free settlement eventually outpaced convict transportation, and it followed a different geographic pattern entirely. The Australian gold rushes of the 1850s pulled in migrants from across the British Isles who had no connection to the penal system at all, drawn instead by the same economic ambition that sent people to California around the same decade. Scottish emigrants in particular arrived in large numbers during this later wave, often settling in Victoria, and their descendants today show up in the genetic data as a distinct layer sitting on top of the earlier convict-era ancestry, evidence that Australia's British genetic inheritance was built in at least 2 separate historical waves, not one continuous migration. Canada tells a related but distinct story, because British settlement there overlapped and competed with French colonization for control of the same territory. English and Scottish migration into Canada accelerated sharply after 1763, when Britain gained control of former French territory following the 7 Years' War, and again throughout the 19th century, as economic hardship and land scarcity pushed hundreds of thousands of Scots and English settlers across the Atlantic. Genetic surveys of Canadians today show a population where British ancestry remains the single largest European component nationally, though it coexists, sometimes uneasily, with a large and culturally distinct French-descended population concentrated in Quebec, a genetic and political fault line that has shaped Canadian identity for more than 250 years. South Africa presents a genetic legacy that is smaller in absolute numbers, but historically consequential in a different way. British colonization there, beginning with the seizure of the Cape Colony from the Dutch in 1795 and solidified through the 19th century, brought English-speaking settlers who established themselves alongside, and frequently in conflict with, the earlier Dutch-descended Afrikaner population. Genetic studies of white South Africans today can often distinguish between British-descended and Afrikaner-descended ancestry with real precision, a division that mattered enormously in South African political history right through the apartheid era and beyond, when English and Afrikaner descendants occupied different positions within the same racial hierarchy, despite both tracing back to European colonization. The two Boer Wars, fought between the British Empire and the Afrikaner republics at the turn of the 20th century, hardened that genetic and cultural boundary into something closer to two separate settler nations, sharing a single country. A division whose genetic outline researchers can still trace today with more precision than most South Africans realize. India offers perhaps the most surprising chapter, because British genetic influence there was always numerically small, even at the height of imperial control. The British Raj governed hundreds of millions of Indians directly, yet the actual number of British colonists who settled permanently and had children with the local population remained comparatively tiny, a reflection of both distance and, especially after the 1857 rebellion, deliberate policies discouraging intermixing between British administrators and Indian society. What genetic traces do exist tend to cluster in specific communities, most notably the Anglo-Indian population, descended from unions between British men and Indian women, primarily during the earlier, less rigidly segregated decades of the East India Company's rule in the 17 and early 1800s. Anglo-Indians developed their own distinct cultural identity, English-speaking, often Christian, occupying a social position that neither fully British nor fully Indian society entirely accepted, and that ambiguous status shaped where their descendants ultimately settled after Indian independence in 1947, with many emigrating to Britain, Australia, and Canada, rather than remaining in a newly independent India that offered them an uncertain place. That 1857 turning point deserves more weight than it usually gets, because it changed the entire genetic trajectory of British India. Before the rebellion, intermarriage and long-term unions between British men and Indian women, while never fully embraced, were common enough that a recognizable mixed population had begun to grow across major colonial cities. After the rebellion was crushed, colonial authorities actively discouraged such relationships, imported larger numbers of British wives to reduce the perceived need for local unions, and enforced sharper social boundaries between rulers and ruled. The genetic data reflects that hinge point almost exactly, with Anglo-Indian ancestry concentrated overwhelmingly in lineages that trace to the pre-1857 period, and comparatively little new mixing detectable in the century that followed. The Caribbean carries a genetic history shaped by an entirely different colonial mechanism, one inseparable from the transatlantic slave trade. British colonies across the Caribbean, including Jamaica, Barbados, and Trinidad, imported enslaved Africans in enormous numbers to work sugar plantations, and the genetic legacy of British colonization there is consequently layered directly on top of and frequently entangled with African ancestry, rather than existing as a separate settler population. Genetic studies of Caribbean populations with British colonial history show measurable European ancestry, disproportionately tracing to England and Scotland, embedded within genomes that are predominantly West and Central African in origin, a direct biological record of the coercive proximity between enslavers and enslaved people that plantation society enforced for more than 2 centuries. Scottish ancestry appears with particular frequency in Caribbean genomes, a reflection of the disproportionate number of Scots who worked as plantation overseers, merchants, and colonial administrators throughout the British Caribbean, often recruited specifically because economic conditions in Scotland itself, following the Highland clearances and repeated periods of famine, had left comparatively few opportunities at home. The United States, despite gaining independence in 1776, remains genetically the largest single repository of British ancestry outside the British Isles themselves, simply because of the scale and duration of English, Scottish, and Welsh migration across the colonial and early national periods. Genetic surveys consistently find British ancestry components present in varying proportions across the large majority of white Americans, concentrated particularly heavily in New England, where the earliest and most sustained English settlement occurred beginning with Plymouth and the Massachusetts Bay Colony in the 1620s, and in Appalachia, where a distinct wave of Scots-Irish migration throughout the 1700s left a genetic and cultural signature still detectable in the region today, in dialect, in music, and in DNA. That Scots-Irish migration deserves its own attention because it represents one of the more overlooked chapters of British genetic expansion. Beginning in the early 1600s, the British Crown encouraged Protestant Scottish settlers to relocate to the northern Irish province of Ulster, displacing much of the existing Catholic Irish population in a colonization project explicitly designed to secure British control over Ireland. Roughly a century later, economic pressure and religious tension pushed large numbers of these Ulster Scots onward again, this time across the Atlantic to America, where they settled heavily in the Appalachian backcountry throughout the 1700s. Their descendants, sometimes called Scots-Irish or Scotch-Irish Americans, now number in the tens of millions, carrying a genetic signature that reflects two successive migrations rather than one, first from Scotland to Ireland, then from Ireland to America, each leaving its own trace still legible in the genome centuries later. New Zealand's story runs closely parallel to Australia's, though on a smaller demographic scale. British colonization there, formalized through the 1840 Treaty of Waitangi with the indigenous Maori population, brought predominantly English and Scottish settlers whose descendants make up the majority of New Zealand's population today. Genetic studies show New Zealanders of European descent carrying a British ancestry profile broadly similar to Australia's, though with a notably higher Scottish component in certain regions, reflecting specific 19th century settlement schemes that recruited disproportionately from Scotland, particularly the Otago region in the South Island, which was founded explicitly as a Scottish Presbyterian settlement in 1848. Beyond these major settler colonies, smaller but genetically distinct British communities took root in less commonly discussed corners of the former empire. Kenya's highlands attracted a wave of British settlers in the early 20th century who established coffee and tea plantations, leaving a small but genetically traceable population still present in East Africa today. The Falkland Islands, settled almost entirely by British colonists beginning in the 1830s, remain genetically one of the most concentrated pockets of British ancestry anywhere outside the British Isles themselves. A tiny population whose isolation preserved a genetic signature nearly untouched by the mixing that diluted it elsewhere. Even Hong Kong and Singapore, governed by Britain for well over a century as trading and administrative hubs rather than settler colonies, developed small but historically significant Eurasian communities descended from unions between British merchants and administrators, and the local Chinese and Malay populations, communities that, much like the Anglo-Indians, occupied a distinct social category all their own under colonial rule. What ties all of these regional stories together is a pattern researchers didn't fully appreciate until they could compare the data side by side. British colonial genetic influence wasn't evenly distributed across the empire. It clustered heavily in what historians call the settler colonies, Australia, Canada, New Zealand, the United States, and to a lesser extent South Africa, places where British migrants arrived in large enough numbers and stayed long enough to become demographically dominant. In colonies governed primarily for extraction rather than settlement, India being the clearest example, the genetic footprint remained comparatively small, no matter how total the political and economic control actually was. That distinction matters, because it means the genetic legacy of British colonization and the political legacy of British colonization don't map onto each other in any simple way. Political control over India was total for nearly 2 centuries. Genetic influence there remained modest, concentrated in specific mixed communities, rather than spread broadly through the population. Political control over parts of the Caribbean was similarly comprehensive, yet the genetic legacy there is inseparable from, and numerically smaller than, the African ancestry forcibly brought to the same islands. Meanwhile, in Australia and North America, comparatively brief periods of intensive settlement produce genetic dominance that persists, essentially unchanged in its broad outlines, more than 2 centuries later. These findings come with the caveats that apply to all large-scale ancestry research. Reference panels for the British Isles, however detailed the 2015 study made them, still can't perfectly capture every historical population that has since moved, intermarried, or been displaced. Commercial genetic databases draw disproportionately from people wealthy enough and interested enough to purchase testing, which skews samples in ways researchers have to account for statistically. And ancestry percentages, particularly smaller ones representing more distant or more mixed contributions, carry real margins of error that widen as the events being measured recede further into the past. None of that undermines the broad pattern these studies have independently confirmed across multiple research teams and multiple countries. It simply means the precise percentages should be read as well-supported estimates, not exact figures. What emerges from all of it is a genetic map that looks almost nothing like the political map history books draw of the British Empire at its height. The empire's political reach was genuinely global, stretching from Canada to India to Australia to countless smaller territories in between. Its genetic reach was global too, but unevenly so, concentrated most heavily wherever British migrants actually settled in large numbers and had children who stayed, rather than wherever the Union Jack simply happened to fly. Consider what that means for the roughly 1.5 billion people alive today in countries with substantial historical ties to British colonization. Millions of white Australians carry a genetic signature traceable to specific English counties, their convict or free settler ancestors left behind 2 centuries ago, whether their families know the details or not. Millions of Canadians carry British ancestry that has coexisted and occasionally collided with French colonial ancestry for 2.5 centuries. Millions of Caribbean descendants carry Scottish and English ancestry inseparably woven into genomes shaped primarily by the forced migration of enslaved Africans. And scattered smaller populations, Anglo-Indians, and Hong Kong's Eurasian community among them, carry a British genetic thread that colonial society itself was never quite willing to fully claim. Genetic ancestry and national identity, once again, don't map cleanly onto each other anywhere this pattern appears. An Australian whose family has considered itself thoroughly Australian for 6 generations may carry a genome that traces with striking precision back to a single English parish. A Jamaican family that has always identified simply as Jamaican may carry a Scottish surname and a Scottish DNA signature dating to an overseer who arrived on a plantation ship in the 1700s. Genetics can identify where the ancestry originated. It cannot dictate how the descendants of that ancestry should understand themselves today, generations removed from an empire that no longer exists in any political sense at all. Return to that woman in Sydney, staring at a result that pointed to a Yorkshire parish she'd never visited or even heard named by anyone in her family. Her identity as Australian was never in question, and the DNA didn't change that. What it did was fill in a specific, previously invisible detail in a much larger, largely untold story, one shared by tens of millions of people across 5 continents who carry some fragment of the same imperial migration inside them, whether their family ever spoke of it or not. Long before any genetic test existed to trace it, and long before any historian sat down to map where the empire's political reach and its genetic reach actually diverged, the migrations themselves had already happened, ship by ship, generation by generation, across every ocean the empire ever crossed. The DNA never needed an empire to still be standing in order to remember exactly where it had been. It simply waited in genomes scattered from Sydney to Bridgetown to Toronto for a science precise enough to finally read the map the empire itself had left behind.